Hola, hola, buenos días. Buenos días a todos. Un gusto nuevamente empezar con nuestro tercer seminario. Este seminario va a ser en inglés, por lo cual comenzaré a hablar en inglés. Tenemos invitados desde China y Estados Unidos. Hello, good morning to Chilean people. Also for the international audience, we will start our third seminar. Before I start, I want to show you my screen. Okay. You can see the program for this morning. We had two speakers from first one is the Dr. Lin San from Beijing Normal University and Dr. Andrew Ogram from University of Florida. Okay. Before I start, I want to explain what is the webinar. This webinar is made with Applied Microbial Ecology of La Frontera University. It's try to understand how to say the title, understanding the role of the bacterial community in anthropogenic impacted aquatic ecosystem. Okay, in the first we had two seminars in Spanish and two seminars in English. Yeah? The first two seminars in Spanish we already finished. Is in August. In August, we had very interesting presentation, but in Spanish, and then we start our third session of our seminar. How to say? We had Dr. Lin and Dr. Ongra from like a collaborator, like a invited speaker. Okay, we hope everything is okay and we can share with you the presentation. Okay, but the first presentation is made with Dr. Lin Shang from Beijing Normal University, and the presentation is distribution, transfer, enrichment, and ecological reach of common antibiotic in water sediment of shallow lake and north of China. And then continue with the presentation of Dr. Andy Ogram. The title is Intersection of Nitrogen and Sulfur Cycles in a Mexican cenote with the implication for export of nitrogen to the sea. Okay, and finally, we make a presentation about one of the main projects is to develop a harmful algal bloom monitoring method and forecasting system for sustainable aquaculture and coastal fishery in Chile. Despite this presentation is focused in freshwater environmental, I include the last one presentation because I think algal bloom is not restricted only marine water. Also, is a river or lake. That's very important. We seminar is focused in the lake of the South Chile, like a protection and to prevent contamination. Okay. Before to finish, also I want to remember you. We had the fourth session of this seminar. We had very interesting presentation in the next September 20. We had a Dr. Xiao Rong from Fuyu University from China with the presentation of glyphosate concentration and bacterial community composition in sediment sample of Yandian Lake. And then we had a Dr. Caitlin Gray from University of Delaware, USA, and the presentation is effect of salinity and biochemical cycling of phosphorus in coastal soil. Okay, and finally I want to share with you a young collaborator, Dr. Marcos Campos from La Frontera University, and show the last data he obtained from a study in the lake here in our region, near to my city, Temuco. Yeah, and the title of that presentation is Spatio-Temporal Distribution of Phosphorus Fraction in relation to phosphormonosterase activity and bacterial phosphormonosterase genes in surface sediment of Bude Lake affected by anthropogenic activity. Okay, before continue, also want to say this, we had the support from Chilean Science and Technology Agency. We had continued collaboration with international project with USA and with China. That is, we can the chance to collaborate with the scientists from different countries. That's very happy. And thank you for your cooperation. I thank you for your assistance. I want to say also cooperation to Dr. Andy Ogram and Dr. Lin for stay here in the presentation. Okay. Before to continue, I want to introduce you Dr. Link. Okay. Okay, now I'm back to the software. I went, please, Claudio, please show to Dr. Link, please, in the screen. Okay, hello, Dr. Link. Thank you very much for staying in this seminar. Thank you for your collaboration. You can start to share your presentation for start to talking. Okay.
Okay. Thank you, Professor Milka. Hello, good everyone. I'm Zhang Ling. Come from Environmental School, Beijing Normal University, China. It's my pleasure to share our research. My topic is distribution, transfer sources, and ecological risk of common antibiotics in water sediment of large shallow lakes in North China. And I will share our research from this.
Please, can you share the presentation? Or Claudio, you can share the presentation? Okay.
Did you see?
Okay. Now it's okay. Thank you.
Okay. Okay. I will share our research from this part and finally conclusion our research. And the first part, I will introduce our research background. And antibiotics are widely used in medical treatment, agricultural breeding, and other aspects. This super performance has bring huge benefit. Most antibiotics in gas, human, or animals are not fully absorbed and metabolized, but enter environment through extra or another ways. Antibiotic residue in the environment will produce selection pressure on microorganisms, make them resistant to antibiotic, become a new environmental pollution. New antibiotic resistance genes, which is a new pollution first proposed by Pruden in 2006. Overuse and improper use of antibiotics in environment has attracted widespread attention. And 2070 is the 19th World Hand Health Day. The Chinese theme is everyone's responsibility to resist antibiotic resistance. So this means that contamination and related problems caused by antibiotics are received increased attention. And through literature research, we found the following widely concerned antibiotic issue, such as waste water treatment plant change discharge antibiotics to environmental, the antibiotic deposition, and antibiotic with a heavy metal relationship. And someone proposed we should impose risk assessment study, or we should understand the factor and driving machines in wastewater habitat. And in our country, someone to calculate the total use of these 76 chemicals in 2013 was 92,700 tons, of which an estimated 15,400 tons antibiotics were extracted by human and animals. Finally, to receive the environmental with 53,1,800 tons of antibiotics. And this list is the number of study detected number of each classes of antibiotics in global lakes, and they found all lakes experienced antibiotic pollution with high detection frequencies of sulfamidol and tetracycline. And in our country, maybe 39 antibiotics are detected in the adequate environment of lakes. And QMS antibiotics are predominant risk and pollution factor in adequate environment of lakes. And then We focus on the lake and why? Because water is one of the important storage places of antibiotics in the environment. Studies show that human activities emit antibiotics with livestock raising and agricultural wastewater discharge, such as hospital water, wastewater, and damaged sewage, as well as agricultural drainage. The sources of antibiotic pollution in the water environment include above these sources. They discharge to environment will accumulate along the food chain and the food web in the environment, and the ecological impact will become more and more obvious with the accumulation levels. Studies have shown that some antibiotics present a high risk for antiquate organisms, such as fish or aquatic plants. Antibiotics also easily linked to the emergence of drug-resistant bacteria in water, such as ARGS. Therefore, antibiotic pollution in water has been widely concerned in recent years. And there are three paths we focus. The transfer of antibiotics in different and the possibility, the possible exploring ways to human health is different. Our research focuses this aspect in water sediment system. The sediment, first, the sediment of pollution from water. Second, the resuspension of pollution from sediment. Third is antibiotic accumulated by aquatic plants. Therefore, based on the pathway of these antibiotics in the water sediment system, we want to understand the accuracy, distribution, transfer, and enrichment characteristics of 9 common antibiotics in BYD, as well as risk assessment and sources analysis. This work will help us to gain an insight into the contamination and the risk characteristics in lake system. Many times provide a basis to propose proportion mitigation strategy for antibiotic management in similar lake systems around the world. And then I will introduce our research area and a simple site. The geographical location of the BYD is shown in the picture. For nearly half a century, due to the comprehensive effect of climate change and human activities, the Bayandin Lake has affected problems such as insufficient water sources, shrinking wetlands area, water pollution, sediment accumulation, and reduction of biodiversity. Water pollution has affected the industry and agricultural production and urban living water safety in Bayandin Basin, leading to the decline of its ecosystem, serving function, and other crises. The most important sources of extra pollution is come from this way, which transport domestic sewage and industrial wastewater from Baoding City to Bayandin. Baoding is located here. The main internal pollution sources are agriculture, resident disturbance, upstream wastewater. Okay. In our study, we selected 9 antibiotics of 4 types common antibiotics in Bayandin Lake as research object. The antibiotic is SAS, include SDZ and SPD. TCS include TC and OTC. QNS include CIP, NOR, and OFR. MIS include ERM and ROM. The physical and chemical properties are shown in Table 1. In addition, 61 sample sites were set up in the whole Baiyangdian area based on the field surveillance result. According to the physical and chemical purity of water sample and sediment, 61 sample points were divided into 5 functional area according to the result of cluster analysis, which show as these 5 functional area. In below introduced present, we will use the aberration. And second, we will introduce the antibiotic spatial distribution. In the whole BYD area, the concentration of NOR was the highest. Is more than 2,500 nanograde per liter, followed by OTC and TC. This is because NOR is primarily used in agriculture, while OTC and TC are primarily used in livestock production. And in the past decades, BYD relied on the abundance and convenient water resource to carry out agriculture and develop the livestock breeding industry. Thus result in high content of NOR, OTC, and TC in Baiyangdian. In addition, the upstream urban sewage wastewater into Baiyangdian through the Fuhe River result in high content of these antibiotics. The spatial distribution of antibiotic contents in overlying and the surface water showed that most areas contained higher total antibiotics content, while only a small portion of the sediment and pore water contain high content antibiotics in here. Above is the whole Baiyangdian area content of the antibiotic, and now we'll introduce in the 5 functional area the content of the antibiotics. The spatial distribution of the total amount of 9 antibiotics in 5 functional areas shows that RF region was the highest, followed by LR regions. This may be due to high levels of antibiotics left over from past agricultural activities in RF area. LR area serves the outlet of the whole lake area, carry out pollutants into the LR when Fuhe River flows through villages and fragments fields which led to accumulated antibiotics. In each of 5 function regions, pore water contained a higher total amount of antibiotics, and the total amount of QNS and TCS was higher at each sample site. We compare antibiotic in global lakes through literature research. The concentration of 9 antibiotics in the 4 types we focused on were 10 to 100 ppt in the water and the sediment. However, only SPD is less than 10 ppt in water and close to 10 ppt in sediment. The content of BYD antibiotic may be slightly higher than this taken. Probably because antibiotics were widely used in China in the past due to the easily availability and its price is low. This is in the surface water, 9 antibiotic spatial distribution. So in this part, we conclude the spatial distribution of antibiotic content in overlying and surface water showed that most areas contained higher total antibiotic content, and the total amount of NOR, OTC, TC was the highest. Compared with functional regions, the total amount of antibiotic in RF and LI regions is highest. And through transfer and by accumulation. And in this section, we will introduce antibiotic in binding, how to transfer, and by accumulation. First, let's look at the migration transfer of antibiotics in water and sediment system. In this part, we analyze the transfer of antibiotics from sediment to surface water, overlying water, pore water, and the whole water body respectively. In the whole BYD region, the partition coefficient log KD of the antibiotics of TCS and MCS type are relatively higher. Regardless of the distribution of sediment to surface water, overlying water, or pore water, SNS has the lowest partition coefficient, which may be because it contains two ionic functional groups, aniline groups and amide group, which has a weak absorption in soil and is easily to migrate into water. This is the LB area. In this area, the partition coefficient of the overlying water and the surface water is obviously different from that of sediment and pore water. In particular, partition coefficient of the sediment to water were not all less than 0, and CIP, OTC, and ERM showed higher than other antibiotics. CIP, OTC, and ERM in the environment are mainly discharged into the environment when domestic sewage due to the absorption then accumulation to sediment. However, the interface between sediment and overlying water is prior to hydraulic and biological disturbance, and the substance is released again. So this shows a higher disturbance coefficient in this picture. And then in UR area, partition coefficient of UR was very special. Most partition coefficients show less than 0. This may be the nitrification reaction degrade antibiotics, and we can say that by the physical-chemical properties, the nitrification reaction since UR area was stronger than other area. And in the NR area, logic KD of NR shows similar regularities between surface water and overlying water, pore water, and sediment water or sediment. Moreover, partition coefficient of the MNS were all more than 0. The adsorption capacity of MNS in sediment is weak. It may be related to the small amount of these active groups in the molecule per unit mass. And in the CS area, partition coefficient of CS still shows surface water, overlying water, and pore water, sediment. These four parts are different. Maybe the oxidation reduction potential of sediment in the CS region is higher than that other regions. The dissolved oxygen in surface and overlying waters is highest. It's account for the difference. And in the NF area, OF showed a distinctive partition coefficient, which are all greater than others. Than sediment and surface than water are most greater than 0, more than 0. And OFL as a step of QS antibiotics tend to adsorb in sediment. easily easily to photolyze in water, and the photolyze rate is related to dissolved oxygen. And now let's analyze the bioaccumulation of the BID. And the BID area phragmites is an advantage plant of the BID and also an economic plant in the past. In our study, phragmites samples were collected to analyze its enrichment effect on antibiotics. In the whole Bayandian area, and SIS type antibiotics have a higher BIF. This is SIS antibiotics. And then we will analyze from 5 functional areas. First, the LB area. In the whole BID and SIS antibiotic have higher BIF, which may be due to the fact that SIS are easily dissolved in water and not easily absorbed by the sediment. And in LB area, BIF in surface water is higher than others, which probably due to the deeper water and the vigorous root and term structure of phragmites. There was the surface water. And in the UR area, the BIF of SIS is still the highest, but the BIF of the CIP of the CIP, TC, OTC, and ERM in surface water, overlying water, and pore water is higher than that sediment. Concentration of antibiotics in surface and overlying water is less than in pore water and sediment in GI region. This may be the reason. And in LR area, the BIF of antibiotics in surface and overlying water is generally higher. The upstream to the downstream by flow water and easily absorbed by particles. Hydraulic disturbance linked to it cannot be deposited, so it is enriched in the surface and overlying water and absorbed by plant. And in CS area, the BIF of SIS are consistent with that previous, still is higher. And BIF of CIP and the TCS were higher than other function area. The OIP in this area is highest, which may be affected by the occurrence and the degradation of antibiotics and further affect the enriched on place. And in the IF area, BIF of N1, CIP, and OTC were higher than others in surface and overlying water. N1, CIP, and OTC are widely used for livestock and agriculture because they low price. The resulting wastewater and manual is applied to field on treatment. Result in residues that are then enriched by the plant. And in this part, we conclude that the partition coefficient of sediment overlying water was higher in the whole BID, is Bayandian, and in different functional areas, especially QNS antibiotics. In terms of bioaccumulation, the water soluble SIS showed a higher enrichment level. BIF of antibiotics in surface water and overlying water was higher than that in sediment and pore water. And for sources analyzed, sources analyzed in this part, we use the PMF model to analyze the sources of antibiotics. And first, PC analysis was conducted to determine the number of factors that could be explained the maximum extent. Through analysis 4 factors should be selected. Then the optimal scheme is calculated according to the PMF models. We analyze the sources of sediment, surface water, overlying water, and pore water. The results showed that first we analyze the surface water. The result is the factors, 4 factors, the loaded take up a large proportion respectively. That is the CIP, ROM, SIS, and TCS, and NOA. According to the main uses and emissions of the antibiotics, the main pollution sources are determined as a finger. The 4 sources is domestic drainage, and hospital discharge, and livestock, and agriculture. And in the overlying water, we analyze the result is SPD and TCS are the most loaded, allows 4 factors, SDC and EM, LM, QNS. Therefore, the sources of pollution were identified as livestock, domestic sewage, hospital wastewater discharge, and agriculture. And this is sediment and pore water sources analysis. Sediment and pore water sources analysis are similar, except for ROM, because ROM is type of SIS antibiotic, and it's easily hydrolyzed, so the content may be low compared with sediment. Therefore, ROM loads in sediment and pore water are different. ROM. So in this part, we conclude that extra sources import under the structural characteristics of the antibiotics. The physical and chemical characters of water and sediment cause the source of antibiotic in surface water or overlying water. Pore water and sediment different load in each factors. The sources of antibiotics can be broadly divided into domestic drainage, hospital discharge, agriculture, livestock breeding. And 5, risk assessment. Antibiotic with the highest environmental risk should be monitored for risk assessment to provide recommendation for police maker and the manager to prioritize the control of antibiotics with high harmful effect. Under certain conditions, antibiotic in water, in sediment, or in our organism can cause environmental risk. So we depend risk quotations, IQ, was estimated based on the report predict no effect, and to divide into 4 degree aspect is no risk, low risk, mid risk, and high risk, and then to assess the antibiotic risk. This picture is a risk assessment of antibiotics in sediment of the Dongting Lake in China. Showed that high risk of antibiotic is a CIP, low risk of antibiotic is TC and OTC, and middle risk is SPD, this antibiotics. And in our assessment result is the IQ of overlying 板垫 surface and overlying water is between 0.1 to 1, which is a middle risk. And IQ NY in BYD poor water is much higher than more than 1. Indicator is high risk, and in sediment the risk is between 0.3 and to 0.4, which is a moderate and moderate risk level. And we analyze risk from sediment, and the result is IQ of NOI in sediment was greater than 0.1, and indicator is middle risk, while others risk are low. This active group contribute greatly to absorb our Q means NOR antibiotics in sediment and learned to accumulate on the surface sediment. And About surface water risk assessment as the IQ of NI is rated 0.1 to 1 in surface water, which is middle risk still. And however, in different functional area, the risk assessment, the result is different. UMRF and CS regions showed low risk values for IOM, which may be due to the inflow of the river in this region, which caused the increased IOR risk. And in the overlying water risk of assessment result is the IQ of NR maintained the same middle risk as other areas in the overlying water. While the risk value of IOM and SPD in LR, UR, and CS region also increased. Not only because the oxygen external introduced of rivers, but also because their structural characteristics, the soft capability in sediment is weak and easily to release into the water body. Finally, when assessment risk poor water, the IQ of the poor water present high risk in all functional area. This may be NI is easily adsorbed in sediment and is released due to the inflow's physical chemical factors such as pH of sediment. So it shows a uniform high risk level in poor water. In this section, I will conclude NI showed middle risk levels in surface water, overlying water, and sediment of each functional area, and extremely high risk levels in poor water. SPD and IOM also showed middle risk levels in external sources in water. Finally, we conclude our research as first, higher concentration were found in BID, NOR, OTC, and TC. The concentration is high. The total about 9 antibiotics was the highest in RF and LI regions. Third, second, most antibiotics cause a high partition coefficient between overlying water and sediment. And third, the BF of SLS with good water solubility was the highest. BF of the antibiotics in surface water and overlying water was higher than that in sediment and poor water. The main sources BID antibiotics are domestic drainage, hospital discharge, aquaculture, and livestock breeding. And NOR has high ecological risk, which is extremely high in poor water, should be focused. And in the future of research, we will focus on one antibiotic. Okay, thanks for your attention.
Hello. Thank you,
hello.
Dr. Lindsay. Very interesting presentation about the antibiotic in the lake water. They had some question for you here in the chat. Maybe you can see in the left, right. It's a comment.
Okay.
I will read. Okay. This is Dr. Chang. Are there differences in the half-life of the antibiotic in the sediment compared with the surface waters? I would like to know if physical disturbance increase their bioavailability.
It means in surface water, overlying water, and poor water, the antibiotics is different. Our result is in poor water, antibiotic content is highest than the overlying water and the surface water.
The poor water to obtain a higher
concentration. Poor water has, yes.
Okay. And then another question is very interesting. You research, Dr. Chang. How do you assess the spectra of ATP present in water sediment waters? Did you use HLPC, mass, or another technique?
What means how to assessment the different antibiotics risk assessment? Or
Yeah.
as my present, we based on risk. Is this we can show our present.
Okay. I think this question about the technique.
This, yeah, this we based on the risk cutaneous and IQ. And this indicator calculator we use to POEC. This we found in the literature.
Okay,
and to calculate the different antibiotic risk levels.
Okay, thank you. Another question is, what is the better strategy to know the main ATP group, antibiotic group present in the sediment water?
What ATP?
I think I ate antibiotic. No? The best strategy to know.
ATP has ADP.
I think it's an antibiotic, ATP.
ATP, I calculate.
I think the question is, what is the better strategy to know the main antibiotic groups present in the sediment waters? How to detect the main antibiotic group?
This still is about antibiotics in different antibiotics assessment.
But ATP, I think, means antibiotic. That's abbreviation.
ATP and
Huh? Yes.
I can't understand the question.
Yeah. But I think when you start to work, you choose different group of antibiotic. Okay? How to use this group, or how to this group, you have to study.
In our research, we choose 5 functional area. The question is how to choose this 5 functional area. First, we survey in whole BYD area, and we choose 61 sites, and we collect the sample about water sample and sediment sample, and to analyze their physical and chemical properties, and to collateral analyze. And collateral analyze the result is to divide 61 samples to 5 functional regions.
Okay. And also, I have a question for you. Is you in the final you measure the risk, okay? High risk, low, medium, or low risk. It's in the case how result or you make a qualification between different antibiotics, but this risk is taken by the environmental office in your country. I mean, the government or some environmental agency take your result and make some measure about that. Talking, it's transferred your result to the society.
Yeah, our result about risk assessment in the poor water antibiotic has a highest risk.
But the people, you transfer this result to the people or to the government to show what we have to do. The risk is high. The risk is high. What we have to do for the people. Not yet.
What we should to do is actually recent years, especially recent years, in BYD area, every winter and summer, government will to remove the sediment in BYD area. Yeah, maybe sediment accumulate a lot of pollution, expect antibiotic, they accumulate another pollution, heavy metal, others. So they remove a sediment to another place.
Okay,
Yeah,
that's a measure to prevent the risk. Okay, thank you. But we had a similar, we don't have so much study in Chile like that. We had to improve in that. Like you had a very good different site, different antibiotic measure. But thank you very much for your presentation. And then you can keep there because now is we check is with the Dr. Andrew Ora from Florida. I don't know, is already connect with us. If okay, hello,
Hi,
Andrew.
no, hello.
How are you? But I want to talk to the people, to the audience. We had some little problem, technical problem with Dr. Andrew. Can you fix the problem?
Yeah,
You can share.
I believe so.
Okay, please, you can share your presentation. Thank you, and of course, thank you for staying. Okay, well, nice. We can see your presentation. Now is your presentation in the PowerPoint. If you put in the presentation mode, it's better.
Yeah, just a second. Hang on.
Okay.
Just a second. Just a second.
Yes. Am I clicking the button?
Okay.
Okay, nice. Thank you. Yeah, please go ahead, Andrew. Thank you very much for your attendance to this seminar.
Okay, thanks. They decided to mow the yard right in back of me, right outside the window. So if you hear some buzzing, that's what that is. So what I'd like to talk about today is some work that we did a couple of years ago on the intersection of the nitrogen and sulfur cycles in a Mexican cenote. And there are some very clear implications for export of nitrogen to the sea. So nitrogen contamination and eutrophication are a major issue in North America anyway. So this is the guy that did all the work. This is Libin Huang. Libin is familiar to at least some of our Chinese friends, like Dr. Xiaorong and Dr. Bai. This is Libin sampling in a cenote. So I'd also like to say that this work was just recently accepted for publication in Environmental Microbiology. Okay, so to give you an idea of what we're looking at, we're really studying the transport or the export of nitrogenous wastes and seawater from inland to the sea, and this is a cross-sectional diagram of what we see. So what we see, this is the land, and this area here is the sea, and so we have an area that's a cenote. In this area of Mexico that I'll show you in a minute, fresh water is exclusively in groundwater. So it's a really interesting system where you have very, very high hydraulic conductivity rates or percolation rates of water. And all of the water that falls through high rainfall goes into groundwater. And the easiest access to the groundwater is in sinkholes or cenotes, as they're called in Mexico. So this represents a cenote or a sinkhole. And it's connected through a conduit from upland into the sea. And then once it hits the sea, it comes out through a spring. So this is a freshwater spring, more or less freshwater spring. There's a salinity gradient in most of these cenotes. This is a particularly deep cenote, but there's a salinity gradient that is fairly low when you're above a chemocline, and then it increases dramatically at an area at some point. And the there are a number of other properties that are right below the chemocline. So at the chemocline, you have high salinities, some exceeding seawater, and we also have low redox potentials and a number of other issues that I'll be talking about. And then they are connected. There are dozens of cenotes in this area that are connected to the sea by these conduits. So we have gradients in oxidative reduction potential and opposing gradients in dissolved organic carbon. What happens is you get nitrogen, largely in the form of ammonium, and dissolved organic carbon running into the water into the cenote through just runoff, and the organic carbon collects as detritus down at the bottom. And so you naturally get a decrease in oxidation reduction potential, an increase in salinity, an increase in density here. When it comes out of the spring, the springs contain high levels of ammonium and dissolved organic carbon, and they also have low ORP or oxidative reduction potential, and relatively low salinity because it's relatively low, because a lot of it is the fresh water that's coming out, and low sulfate. So it's more characteristic of the groundwater than it is of the seawater, as you would expect. So this is more or less a freshwater spring. There's an interesting tidal variation in these areas. If you have high tide, then you have reversal of flow, where you have the salt water that's low in ammonium, low in DOC, high ORP, high salinity, high sulfate, pushing down. And so you have this cycle. It happens twice a day, where you have either fresh water being exported to the seawater, or you have seawater moving in. So that creates a zone in this conduit that's a mixing zone. And that's primarily the area that we're really interested in, because it's probably chemically and biologically reactive. The problem is we can't really sample in that area. To give you an idea of the distribution of this type of phenomenon, where you have groundwater connecting with seawater and a mixing zone below, each of these red dots represents a number, not just one, but a number of different subterranean estuaries. So they line the United States and much of North America. There's some in South America. None have been mapped to Chile in this diagram, but there's one in Peru. Probably this is a very incomplete study. You also see some in East China, but I suspect there are more. And they're all around the world. So they play a major role in coastal nutrient cycling, because this is a convenient area where you get export of nutrients such as ammonium and phosphate out from the terrestrial systems into near coastal waters that can lead to eutrophication. So eutrophication of near coastal waters is a concern. This is what the cenote is that we were studying. We were looking at cenote siete bocas, which means 7 siete mouths into this cenote. And these are just a couple of the different mouths that you can see. But it's really a great place, and it's an area for tourism. So people, not just tourism, but recreation. So a lot of people come in here and swim. It's really pretty cool. So this is where we sampled. We sampled in cenotes siete bocas and an offshore spring, Pargos. We sampled at 20 meters, 28, 29, 30 meters, and 32 meters. The reason that we have a more intense sampling in this area is we wanted to capture the chemocline. So what you have is low density fresh water on the top, and then you have the higher density saline water that's below this area. And so we're interested in the processes that occur in the low sulfate area and low density, and the high sulfate, high salinity areas. We also could not sample this, the conduit, but we could sample what's coming out. And so again, this is Pargos. It's on the outside of the, it's in the near coastal area. It's maybe 100 meters offshore. It has two mouths that come into it, and a left channel and a right channel. And we found out later that they probably do not connect to the same conduit. And we sampled in the mouth on each of these mouths, and about 2.5 meters above the mouth, and then finally up above the area that would be related to the spring. We took those water samples, we filtered them, isolated the DNA, and then used 16S amplicon sequencing. If you're interested, we used Illumina MySeq samples, and it was processed by Chime 2. And we were also really interested in nitrogen cycling, so we did qPCR of various nitrogen functional genes. We also took a lot of physical and chemical parameters for these. We have quite a bit of data, but in the interest of time, I'm not going to go into those data. Just to give you an idea of what the data look like, this is up here. We have the data with depth for C7b, and then this is the spring Pargos. We took 2 different seasons, a dry season and a wet season. Dry season is red, the blue season is bluish. And along this axis we have depth, and along this axis we have the magnitude of the parameters that we're studying. And so you can see as we go deeper in Pargos, we see this dramatic increase in salinity at the chemocline as expected, and then an increase in salinity below that. Same is true for hydrogen sulfide. Ammonium is very, very low, 0, until we get below the chemocline, and that's also follows with oxidative reduction potential or redox potential. Phosphate is very, very low until we get below the chemocline. And then DOC is low, but it really, really increases once we get below. And then this is the ORP, where we see it fairly low until we get above, again, above the chemocline. For Pargos, this is reference water that's away from the spring. This is the mouth of the spring, so this is just above the area where it's mixing, and we see salinity predictably is very constant. At low tide, its salinity is a little bit lower because we're getting more of this influence of the fresh groundwater being able to come out. At high tide, there's an imposition of this hydraulic head that pushes it back, so we see an increase in salinity at high tide. Right? And so hydrogen sulfide pretty much follows all of this. Ammonium as well, phosphate, DOC, and ORP. Okay, so it's a tidal system. That's as far as the outside is concerned, it's driven by the tide. High tide, we get greater influence of the seawater. Low tide, we get a greater influence of the spring of the groundwater. Okay, so this is what the microbiology looks like of the system. This is a stacked bar graph. These are stacked bar graphs of the dominant organisms. This is at the phylum level, and this is at the genus level. At the phylum level, what we see almost predictably at the higher depths of C7b, we see it dominated by proteobacteria. As we get down further into the cenote, we see these pink guys. These belong to the phylum Campylobacterota. So Campylobacter, you might be familiar with those, but they actually represent the epsilon proteobacteria, And I'll talk about them later quite a bit. At Pargos, at the mouth, we also see a real dominance of both proteobacteria and the Campylobacter rota. This is the genera within Campylobacter rota. We're really interested in them for reasons that I'll explain in a minute. But they're the Sulphurivorum and Sulphuromonas are dominating the genera within the Campylobacter rota, particularly as we get lower, as we get down into the cenote at the chemocline, and then below that into the saline area, saline zone. So this is why we're really interested in Campylobacter rota. They are the genera that we're seeing are sulfur autotrophic denitrifiers. So that means that they use sulfur, reduced sulfur forms to provide energy for the oxidation of, for fixing carbon dioxide or carbonate. So they use these reduced sulfur forms to fix carbon, and then that is oxidized to sulfate. And as a terminal electron acceptor, they are anaerobes, so they use nitrate and nitrite, but primarily nitrate, to reduce to nitrogen and nitrous dioxide. So the reason that we're interested in this is because there's quite a lot of sulfur, reduced sulfur species, because of the sulfur cycle in these areas. And there's also quite a lot of nitrate that is being produced in the cenote. So this is a good thing. If these guys can process the nitrate to nitrous oxide, then nitrous oxide is a gas and it blows off. So it modulates or limits the amount of nitrogen that's being exported to the seed. So that's a good thing. This is a RDA redundancy analysis. And the way to read this is the arrows, the longer the arrows, the stronger the correlation, and arrows that are parallel or close to parallel are correlated with each other. Arrows that are opposite in direction are inversely correlated, and arrows that are at 90 degrees to each other have no correlation at all. And so what we see is that Campylobacter rota, these guys that are the sulfur oxidizing bacteria and denitrifiers, are correlated with hydrogen sulfide, or sorry, bisulfide, as you would expect, ammonium, which you would also expect, and I'll get into that more later, phosphate, and to a lesser extent, DOC. They are not correlated with pH, ORP, or salinity. For the genera, we see, again, very similar type associations where we see sulphivorum down here. It's moderately correlated with sulfide, with bisulfide. Okay, this is a correlation. It's not a beautiful correlation with the genes that are related to the Campylobacter rota. So we also, I'm not showing you a lot of the nitrogen cycling gene data. This is SOX-B, it's sulfur oxidation for the Campylobacter rota, and it's not really a very a good set of primers. So I kind of take this with a grain of salt. Actually, I probably shouldn't even be showing this. We also see some similarities or correlations with near S, which is dissimilatory nitrite reductase, sorry, dissimilatory nitrite reductase. That's a key component of denitrification. And this is specific also to the Campylobacter rota. And it's very specific, and it probably doesn't pick up all of the guys, all of the Campylobacter rota. but it matches fairly well with SOX-B for those organisms. And it is inversely, or sorry, it is not at all correlated with sulfate reducers or ammonium oxidizers or some of these other bacterial denitrification genes. So from this, we were able to propose a model for nitrogen cycling in the two systems. So first, we see dissolved organic matter down at the bottom of these cenotes. You know, it falls in from the above, and then it collects down at the bottom. It's metabolized, and we see the formation of dissolved organic carbon. It also is, during this DOM decomposition, it releases ammonium, and so the ammonium can be transported out. We also see at the interface and associated with the Campylobacter rota, we see denitrification. Nitrate going to nitrogen gas and nitrous oxide. That nitrate, we think, is coming from ammonium being nitrified to nitrate. And so the nitrate is increasing. It drifts, filters, diffuses down to the point where we have this, this interface, the chemokline. It's metabolized by the Campylobacter rota to produce nitrogen gas or N2O. By the time it gets to the spring, we have low tide. At low tide, we have low RP, high DOC, and we also see assimilation, because we're seeing some loss, assimilation of nitrogen nutrients for microbial growth at that point. And then at the at high tide, we see this kind of mixing effect. All right. So also at the mouth, we would get more of this Campylobacter rota probably being responsible for denitrification. So to conclude then, Cenotes siete bocas may not be directly connected to Pargos. We didn't really establish that. There are dozens of springs in this area, or sorry, dozens of cenotes in just a few springs. So probably what's happening is the cenote that we studied is one of many that are draining into that, that spring. I didn't show you a lot of the data that makes us think that Cenotes siete bocas is not the only one that's draining into this area. And we don't know for sure that it is even directly connected to Pargos. It probably is, but it may not be, but it's likely to be representative to all of those that do connect to the sea. In our study sites, autotrophic sulfur oxidizers belonging to the phylum Campylobacter rota are responsible for removal of nitrogen via denitrification. Copy numbers of MIR-S and SOX-B, so that's denitrification and sulfur oxidation, representative of Campylobacter rota, were consistent with the general 16S distributions of the phylum. So even though those functional primers were not ideal, we think they are still representative of the ability of those representatives to denitrify with sulfur oxidation. Members of the genera, Sulforamonas and Sulforobum, dominate the cenote, Chemolimnion, and then as we get outside, we see that Archobacter, also a Campylobacter rota, dominates the discharging spring water. So we see a difference in composition. There's still Campylobacter rota, there's still sulfur oxidizers, denitrifiers, but there's a selection of the genera as we go from the spring to the outside. And so we conclude that submarine estuaries such as this C7b Pargos system may provide critical mixing zones for the removal of nitrogen contaminants. And so that ends my talk. I hope I've been in time. So if you have any questions.
Thank you, Andrew. Very nice, very interesting place to study, huh? In Mexico.
Yeah, beautiful. It was really a nice place.
Yes, very nice place. I had a question here in the comment. It's from Marco. It's, dear Dr. Oram, fascinating your results. Could you let me know if you have a background on how phosphorus inputs from the sea impact the cenote? This thinking that sea could be richer in phosphorus than fresh water.
Yeah, that's a good question. I don't know. I believe that phosphate is higher in the cenote than it is in the seawater. In this case, we could go back and I could show you the distributions. We have phosphate concentrations. It's because this area is draining a lot of agricultural land, and so the phosphate is really most of the phosphate is originating in that area. Is in the cenote in this case.
Okay, thank you. Here another
sources are driving the phosphate concentrations.
Yeah. Thank you. Then had another question. What other functional are key in the submarine estuary zones? I think so.
Yeah,
Nitrogen, phosphorus, what another, I think.
Yeah, we looked at a lot of, we looked at as complete a picture of the nitrogen cycle as we could. So I looked at genes related to nitrogen fixation, archaeal ammonia oxidation, bacterial nitrate ammonia oxidation, DNRA, dissimilatory nitrate reduction to ammonium, and ammonium oxidation, anaerobic ammonium oxidation. And it looked to us like the key genes that were at least representative. And we didn't look at processes, and we didn't look at mRNA. So we really can't say what the true processes were. But just based on the sheer numbers of the genes there, we think that these other processes were less important. So ammonification in the bottom to produce ammonium, ammonium and nitrate input from above, from terrestrial sources, from agricultural sources, were leading to nitrification. So nitrification is a critical thing, and denitrification was critical. We didn't see a lot of NIFH or nitrogen fixation, although we did see signals related to cyanobacteria, but we didn't think that those were the dominant factors in controlling nitrogen cycles. We did not look at the phosphorus cycle at all because it wasn't that interesting to us. So sorry, we were mainly interested in nitrogen. We did see some very strong signals related to a enterohemorrhagic E. coli strain, which would have been really interesting to pursue, but they were there in the cenote.
Okay, E. coli, maybe tourists.
Yeah.
We had another question from Fumito Maruyama, from Japan. That's hi, Oran. Could you explain what kind of sampling device do you use? I'm worried about the water turbulence during sampling.
Yeah, we used two. We used a tube, just a pump that was pumping very slowly. We would put it down to specific depths, pump very slowly, first through I think it was a 2-micron filter, inline cartridge, and that led to a 0.2 micron cartridge. And so we sampled the 0.2 micron cartridge. Turbulence is always an issue when you have do that kind of sampling, but I think the slow sampling rates would limit that. That was one way when we went to the mouths of the spring. So we had two different channels leading up to this one spring. And so divers went down and collected with bottles into those springs. In fact, the sampling of our of the cenote was so slow. It was really frustrating to the geochemists that we were working with, because we took many times longer than they did to actually do the sampling.
Okay, thank you. And the last one, another question from Dr. Maruyama. What kind of the species of archaea do you find in your samples? Your sample appears very interesting and contain a lot of archaea in addition to domain bacteria.
Yeah, that's a good point. Dr. Maruyama, good to see you. The major archaea that we saw were the nitrophyters. So I don't recall. I'm sorry, but you have me at a disadvantage. I don't recall what the dominant ones were outside of the nitrophyters. They were probably methanogens, but I don't recall in the cenote. But I honestly just don't even recall.
Okay, we had another question, but we continue with the program. I want to show you my presentation before, and then we had time to continue discussing our main question. Okay, for the audience, please send your question by the social media, and we collect here, and we can read it to the speaker. Okay, I want to thank you to the two speakers this morning, and I want to show you what one of the big projects we had in the lab. We can translate or how we can use this project focus in marine environmental to the lake or freshwater ecosystem. Okay, now I will share my screen. Okay. I think you can see my screen, okay? Well, here. Okay, before I start, you know, this webinar or seminar is understanding the role of bacterial community in anthropogenic impacted aquatic ecosystem. That we choose this title. This time in my presentation, we make a brief introduction of what kind of this seminar. We had, I had to tell you before, we had two seminars in Spanish and two seminars in English. In English is more scientific, but in Spanish more transversal area, like a government and some foundation for civil foundation to people worry about the impact of the human activity in the lake in southern Chile. But in this, I want to make the link with this webinar with the project. We had a big project. It's monitoring algae in Chile. It's a monitoring focusing algal bloom. And this in the lake, we had a problem with algal bloom. Like we saw this project was very interesting to apply also in freshwater ecosystem. Okay, in this project, we made a brief introduction also for this project, and we're talking about the concept, the alluvium, and then the term for hubs. Hub means harbor algal bloom that we call hubs, and the objective, a structure of this project, and a specific object in this project, and the advances or result of this project until now, and then what desirable action post and end of the project. Yeah, this focus. We make introduction of this seminar and then how to connect with this big project. Okay. I made this presentation before in Spanish. That now is I want to share this presentation with the international collaborators or with international audience. We talking about this webinar focusing the aquatic ecosystem, like a provider or ecosystem service. Yeah? The aquatic ecosystem, like a river, water, estuary. You can see the photos in our region. Yeah, this water environmental had a lot of influence in the environmental and the people. This river, like an estuary, provide essential ecosystem service for the people and for maintenance of the quality of life and this sustainability. Okay, for the ecosystem correspond what means ecosystem service. This is a multitude of benefits that nature brings to the society. That's a provision of biodiversity, water regulation, like a climate, water quality, erosion control, cultural, like a tourist and recreation, similar with before Andrew showed with cenote, is very recreational and tourist place, and support the primary production in the ecosystem. Okay, that means is aquatic ecosystem very important in our region. In this science, biodiversity, this is the wide variety or diversity of the living organisms, is essential for the functional aquatic ecosystem and for them to provide this ecosystem service. Okay, that's. But we had, the last few years, we had a problem in Chile because, you know, in our region, increase the population, increase the touristicity, increase the industrial activity, and the city become bigger every year. That's, we had a warming about 3 or 4 years ago. That in this case, we had a big, beautiful lake here, Villarrica Lake, is a declared as saturated zone by chlorophyll, transparencia, and dissolved phosphorus. That is official decree in the government. What means this one? This lake has accumulated nutrients from the air, mainly phosphorus and nitrogen, which is polluted and causes eutrophication in the lake. That's what you see here. In the lake, we had people living, people, industria, and aquaculture factory, and this affect the lake. Yeah? Also, microscopic algae and bacteria feed for this nutrient in the water, on the sediment, and develop explosives that make the algae bloom, preventing the entry of sunlight and consuming the available oxygen in the water and sediment, causing the death or affect many organisms in the lake. Okay, that means its balance and natural purification capacity is broken and affecting the ecosystem service provided by the lake. Now, the government take all this antecedent, all this data, and he declared this decontamination plan is required for the Villarrica Lake. Under this scenario is my laboratory and my colleagues. We had different projects, international collaboration project with China, with USA, and with Japan, to focus in how to prevent this, or collect some information about bacteria community in this water ecosystem or aquatic ecosystem. But why we study community in aquatic ecosystem? Why bacteria? Well, bacteria is one of the most diverse about that group on the Earth, with a long evolutionary history, and they live freely or in association with all other microorganisms like fungi, microalgae, archaea, animals, algae, and plants. They use light and organic compound as a source of energy and source of carbon, and they can live like a symbiont, antagonist, or pathogen. Also, they have to demonstrate, Dr. Oran before, is that an aquatic ecosystem of bacteria can participate in recycling organic matter or carbon, nitrogen, and phosphorus. Is also a source of nutrient for another organism. Also participate in degradation of recalcitrant compounds, and then produce bioactive compounds like antibiotic, hormone, and enzymes. However, its bacteria community are not yet considering the protection, sustainability, and discontamination initiative in Chile. Thus, bacteria is always considered a black hole. Okay, black box. Sorry, black box in this aquatic ecosystem. Okay, that like I show this picture, this can be or come better on knowledge of bacteria community to be used for the protection, sustainability, and the contamination of the aquatic ecosystems of Chile. We can see where do we find bacteria living. The bacteria living associated with different plant, algae, crustacean animals in the lake, also in the column water on the sediment water. That we know very few about this kind of bacteria living in that ecosystem. That there how to we study bacteria community in the ecosystem. We had different strategies. One was, for example, the functional, the enzymatic reactivity, the enzymatic reaction associated with natural recycling. For example, we use a lot study with the use of acid alkaline phosphomonosterasa related to phosphorus recycling, and also makes quantification the bacterial gene is, for example, 60 days for detection for counting total bacteria. Also some functional gene like a phosphomonosterasa gene and nitrogen fixation and nitrogen fixation gene by using, in this case, with quantitative PCR, qPCR that we had. Another technique, we use metal barcoding. That means we take sampling, make DNA extraction, and amplify 16S region of bacteria, and go to the sequencing, Illumina sequencing, and make some bioinformatic analysis, and then analyze the result. What we can do with this sequence information, we can make check the composition of bacterial community, check the diversity of bacterial community, or relation within different bacterial community from this sample. Also, within bacterial community, detect or predict the interaction, and then also function, predict the function of the bacteria. That we can do it with this technique. Okay. Well, we start to talk about the monitoring of algae in Chile. MASH, we call MASH project, is the project. The title is Development of Harmful Algal Bloom, HAP, monitoring method to forecasting system for sustainable aquaculture and coastal fishery in Chile. Okay, this project is a program for science and technology research partnership for sustainable development. We call SATREPS. The abbreviation is SATREPS. Is an initiative supported by Japanese government. In this project, we had scientific collaboration within China, with Chile and Japan. We had colleagues from 3 universities, from Los Lagos, Antofagasta, and UFRO, and for Fishery Institute, IFO, we call. And also we had a collaboration with the Japanese scientists like Dr. Fumito Maruyama from Hiroshima University, and Dr. Shoko Ueki from Okayama University, and Dr. Satoshi Negai from also is a Fishery Institute in Japan, Research Institute in Japan. And this project started in 2018 and finished in the March of 2023. But this project is focusing the hub. Why hub? Because hub is very important for Chilean society. It has a lot of impact in the public health, in the ecosystem, economy, and society. Also, is affect the economy, affect agriculture and the fisheries in the social. That there are many, usually in the summertime, there are many news about this hub, how to have affect to the Chilean society. That's a very priority in Chile also. Okay, but also this big, many Chileans concerned about the hub event, but there are many initiatives, study or investigating hub that we had usually also appear in the news. That kind of initiative. We use the microscope visualization, satellite imaging analysis, and modeling, computational modeling, like that. But what we are different in this project compared to the current initiative in Chile. Yeah, we present here a different alternative. We use the concept holobiome. What means this holobiome? Holobiome means come from the holobiont and produce what do you refer to the host and his microbiota. For example, me, I'm Mirko, I'm person, but also I had a lot of microorganisms associated with me, and these organisms affect my health. Okay, that's we use the concept holobiome like the sum of total of the genome that make up eukaryotic organism. That is the genome of the host, like this means the microalgae, and the genome of associated microorganisms. That we study the microalgae, the hub, like a complex environmental, the interaction within different microorganisms. That's more representative, this slide, what is the determinant of health dynamic. We had the environmental, physical, chemical, and biological factor. We had the light, the temperature, salinity, nutrient current. But also we had the microbial interaction within bacteria, with another microalgae, with the virus, or fires, okay, and zooplankton. We had this interaction that we not study about only one hub species. No, what has species and he associated holobiont. Okay, and what try to looking for? Try to looking for or detect or evidence, find some evidence that some hub microorganisms can promote the hub and other microorganisms can need the hub. That means this only the hub development not only depend the physical chemical factor, also can depend from the interaction, microbial interaction. For bloom, when it start the bloom or when they finish the bloom. But we in this project we focus is that kind of interaction. Okay, that's the main objective of this project is develop a hub prediction system, the prediction system like a early warming, based on hub species monitoring and information accumulation system. We understand the prediction system is a very big goal that any advance about this kind of the study or knowledge about the hub in the Chilean coast is advanced for Chile and for information we can use to prevent mitigate the effect, negative effect of the hub in the society, Chilean society. Okay, the structure is we focus in two kind of the group, the Chilean group and Japanese group, scientific group. Okay, we incorporate here also government office like a Suspesca, Cernapesca, and MISAL here in blue square. Also private industry. We have Intersal is salmon aquaculture, and Intermeat is a viticulture. Okay, that we focus, and then we create the joint coordinated committee with associate and discuss about the advance of this project for not only the scientific point of view, also we incorporate the government and the private sector point of view to discuss. Okay, on the all these organizations is helped JICA, cooperation agency from Japan, from GST is a scientific Japanese science technology agency. Okay, that we had this structure in the project. How to see this is the main objective of this project. Okay, you can see in the four main project, but you can see the arrow is blue and red. Blue means it's a scientific research, and red is a social implementation. That means this project had also a big challenge in the implementation. This transfer the information to the society, private and public sector. Okay, but what is the main objective of this project? The first is identify the microorganisms in the algal bloom by analyzing the structure holobiont of hub, and then to identify the factors that determine the structure of hub holobiont. And third one is to detect the species of algae and bacteria that cause hubs and develop a prediction model. And finally, to establish a platform to share information for the proposals mitigate the associated impact of the hub in Chile. Okay, that's this more discussion within public and private sector. But in the beginning of this project, you use 14 sample sites along the Chile. You know, the most focus in the middle of Chile with the aquaculture is main active, but also we take samples in the north of Chile and the south of Chile, in Antofagasta and Punta Arenas city. Okay, we made the sequence 16S and 18S, measure another parameter like a photosynthetic pigment, qualitative and quantitative analysis of the phytoplankton by microscope, relative abundance of hub species, nutrients, also characteristics of water column, and also its atmospheric parameter. But you can see we affect for the COVID pandemic. That we had to make some arrangement or improve, and the new scenario we decrease for all 8 sample site now. Yeah, for focus more in some sample site, more high frequency of the algal bloom. Okay, the first to identify the microorganism by analysis structure of the holobiont hub, we use a metabarcoding technique using Illumina sequencer, similar to show before Andrew. What is the special of this? Because this no use it globally have a monitoring system or only one system. No using this technique because technique is need more skill and more money for the planet. Now nobody making monitoring or one system based on the holobiome analysis. Yeah. Also, this technique is allowed a greater resolution and determine the composition, the taxonomy activity of holobiome. Also, is allowed a compare similarity and difference between different holobiome of habit species, and also allow inferring the potential function and interaction of components of holobiome. That we only know who is there, also what they are doing there. Okay, we had that potential and who interconnected and interact with each other microorganisms. Okay, now we start from September 2019, and for the June this year we had about 2,000 samples, where most of the samples are already sequenced. And based analysis, you can compare. This is an example. In the left, we compare the description or the structure, the phytoplankton determined by microscope, and we compare with the phytoplankton determined by genetic with Illumina sequencer. And you can see more phytoplankton species, and also we can detect the bacteria in the same sample. Okay, that with all this data, we are generating a public database of genetic information of phytoplanktonic microorganisms and associated bacteria, adherent and free living. That not only this bacteria close living, but also bacteria living around the phytoplankton from the Chilean coast. Yeah, for this 14 sampling site. Okay, in the preliminary results showed the microalgae, some species of microalgae not reported for Chile. It's the first time, and also some specific microalgae in some part of Chile. Yeah, some focusing this. I mean, this microalgae usually is detected in the south of Chile, but we also detect in the north of Chile. Okay, but in the low abundance, but because of this technique had a more resolution that we can detect. Also, there's some bacteria. Even that bacteria is low abundance. There's also some bacteria, some algal species, but producing toxin. That's we created with revising all information about the phytoplankton and associated bacteria in the Chilean coast. With this data, we also now we are analyzing the structure, activity, interaction, that we can check the composition of this with the main family or phylum in this bacteria or algae, also with the relation and interaction within that. For example, in the middle you can see in the left you can see clearly in the violet color the sample taken from the north of Chile, separate from the sample taken from the south of Chile. Okay, that is we can make this differentiation. Each part there's some biogeographical distribution of the phytoplankton. But how to say before, this is important. Not only the information, scientific information, we also want to transfer to the people that we already had the two protocol publication, published protocol, step by step for sampling, sequencing, analysis of happen in Chile. That you can see I imagine you can see that kind of protocol we detect with different point step by step you have to do the protocol, standardized protocol for all Chile. Okay, we had two scientific articles. Now we had a scientific education video. Also, it's in the article you can see the protocol, but easy way also you can see the video how to make the sampling and sequencing analysis. Okay, all this information is free, and in the website you can download, no problem. Or you can ask me, I can send you the link to download. Okay, the number 2 is identify the factor that determine the structure, how to microalgae or bacteria can interact. That's we study that, and we study the interaction between phytoplankton, microorganisms, and associated bacteria on the Chilean coastline by co-cultivation. Okay, inside representative high species and associated bacteria. You can see here. This is a cell well plate. You can see without bacteria, we didn't observe under deficient of iron and vitamin deficiency. We cannot detect microbial growth, but when put some bacteria in under deficient, they can grow. You can see asterisk show the microbial growth. That with only bacteria is helping to the microbial. Then we have selection this kind of bacteria, how to promote this interaction, and looking for make the genome sequencing identification of functional gene, like a vitamin, siderol, hormone, and toxin in the genome. We try to determine what is the functionally, how to improve the microalgae growth, or on the opposite side, decrease the microalgae or affect or kill the microalgae. Okay, the objective number 3 is to detect the species of algae and bacteria that cause HABs and develop prediction model system. In this case, we already developed a suitcase lab. It's a suitcase will include different equipment and material for make the detection of HAB species within 2 hours. Now, this suitcase also is published. You can see the specification or the how to use in detail in this publication. Now it's in the pilot stage. We can adapt. Now we can some specific HAB species detection, but depend on the specific gene functional or a specific species of bacteria or microalgae. We also can modify or use like a gene target for this suitcase. That this portable HAB species detection system in pilot stage that can be adapted to target gene, for example, 16S, 18S, RNA gene, or toxin or interaction gene, and also with target user. The idea is the normal person or a professional technical can use this small device, portable laboratory. Yeah? That we want to use, the people can use it. It's not specific for scientists. Also, we had implemented the experimental stage and advanced portable system and mobile laboratory system. Both of them is for make a study of the holobiome. That we incorporate DNA extraction, sequencer, qPCR, and sampling device and ClinBench to work in the suitcase or in the laboratory, mobile laboratory. That we use not only for science, also we can use for outreach activity, both of this equipment. Also the suitcase number 1, also we can use in different outreach activity. But this different with this the mobile and the suitcase is used for main detection, quantification, activity. That's the main idea. And the previous one is for detect the presence. Okay. Another is develop a prediction model for HAB. Yeah, we now is working with the Japanese colleagues 3 different models about the how to detect. Some holobiome. One is using metabarcoding in data, incorporate in this model. Another is maybe fluctuating interaction network. Yeah, for ADM model is maybe interaction within microorganisms or algae using the private sector database. That both model, but incorporate to now a model prepared or hosted by IFOP Institute, Fishery Institute in Chile, for made the prediction of HAB. Okay. Also, the last objective, this social implementation, is try to do symposium on HAB-related topic, where participation is the private and public sector, training people in using the portable system and mobile laboratory, and also public seminar for to a wider audience. Also, it show the result, this kind of result, analyzed not only for under scientific point of view, but also a Chilean government and private sector industry point of view that try to show the predictive model and discuss with the institution. In this case, we finished this project. We put in the server Shonos. These servers belong to the Institution of Fisheries Chile, IFOP, who is associated to this project. That the Shonos website already had some online system to detection or monitoring the hub in Chile, and we incorporate all our data in this new model in this website. Try to make that system for prediction system for Chile. And finally, the desirable action post-end or project is our data can be useful for the public and for the private sector, and try to improve our detection of warming alert, early warming alert or detection of algal bloom, and decrease or mitigate the effect on the society, on the Chilean society. And this is a big project. We had a lot of institutions here. We can see different the equipment, the senior researchers, associate researcher, and another researcher, outreach coordinator, and administration assistant. Okay, but why incorporate this presentation here? Because we study algal bloom. And how to say before, now the eutrophication in the lake, one of the main indicators is the algal bloom. Appear now, no algal is a cyanobacteria bloom, but we can, this device can be very useful for continuous study the lake algal bloom or the lake impact of human activity in the bacterial community or phytoplankton community in the lake, only in column of water, also in sediment. Okay, thank you very much. I'm here. Sure, continue. Thank you. I hope this presentation very useful for you, and I want to share with you our project, our data. It's now preliminary. We continue. We now is under analysis. We hope the release, some of them release, for example, the protocol and the video you can download freely from the website. Okay. Now, before finish, I want to invite to our speaker, Dr. Andrew Ogram, and Dr. Ling is there, because I don't know if Dr. Ling is still here, because in China it's late. Okay, I think this is only Dr. Ogram, Dr. Andrew Ogram. I had the last question for you, Andy. Okay, or you can also make some question about my presentation too. My question is for Marcos. Is this dynamic could you have implication in potential cyanobacteria bloom episode in the cenote? What is the scale of this phenomenon compared with impacted aquatic ecosystem?
Yeah, I don't know. We detected cyanobacteria throughout the water column in the cenote, from the top really down to the bottom, surprisingly. So through the chemocline and into low redox potential areas. As far as driving cyanobacterial blooms offshore, you would think, I mean, the idea is that by increasing denitrification, you would decrease the amount of nitrogen that's exported. And so you would hope that that would decrease the cyanobacteria, the HABs, harmful algal blooms. But we don't have any data on that. We also don't really have any true data on flux out of it. So Marco's question is, in the cenote itself, don't know. To be honest, you could speculate that it also would probably decrease that potential, but just really don't know. We never observed a bloom. So,
okay.
But that doesn't mean it didn't happen. I don't know.
Andrew, you had a lot of experience in the Everglades in Florida. You in this cenote, you show the Campylobacteriota like a denitrification, no? is present in nitrogen cycle. But in the case of the your expertise in the or your experience in the Everglades, also you can find Campylobacterota or another type of microorganism involved in the nitrogen cycling, for example, the nitrification.
Yeah, so Marco, Milko, I'm sorry, I couldn't understand everything that you said. There's a lot of noise outside, but as far as nitrogen cycling in the Everglades, we found quite a bit of NIFH, so nitrogen fixation, also in areas bacterial ammonia oxidation, but we also saw NMOX going on. Anaerobic ammonium oxidation was also present there, and that's a difference between what we see in the Everglades and what we see in the cenotes.
But you show Campylobacterota. Campylobacterota, this is group. Also is present in the Everglades?
We didn't see it. We've done some work on that, but we didn't see it, at least not in significant proportions. That was dominated by proteobacteria. These are epsilon proteobacteria, but by other proteobacteria. A lot of deltas and gammas in the Everglades.
Okay, thank you. We don't have question for Andrew or for me. We can finish. I think the Dr. Lin is disconnected because China is very late now. That's appreciate to Dr. Lin's participation in this seminar. And also, Andrew, thank you very much for your participation. Next week we had the JC student here, or colleague, also make us another interesting presentation. Okay? Also, I invite you if you will have time to connect. Okay, here is Dr. Link. Thank you. We just finished. Is to say thank you very much for participating like a speaker in this.
Okay, thank you. And there's some problem, and Professor Ba and Xiao want to attend the meeting, but they couldn't attend. The link had some problem, so they advise the next time we use Zoom.
Yeah.
Yeah.
No problem. Okay, I think we have to finish the time. I want to use the time, and we keep in contact. We had, I hope we got good start to collaboration. I had many years collaboration with other. I think when to start the collaboration with Lin Chang. Doctor, thank you very much. We also study the lake. In the next week we are in presentation about the lake. We also try to study some antibiotic in the lake, because also it's important concern here in Chile. Okay, and then keep in contact with you. I thank you very much. Okay.
Okay.
Okay. Bye bye.
Okay.
Thank
Goodbye.
You.
Bye. Bye, Professor Andrew.
Bye.